Good morning everyone. Today we are going to discuss about the polysaccharides. When we talk about the polysaccharides, we talk about the starch, we talk about the cellulose. Then we are starting with the starch. That what is the starch? When we will talk about the starch, what is the starch? We say that the starch is the combination of the two monomers, that is the amylose and amylopectin. When you will see what is the amylose and what is the amylopectin, you will find that the amylose is the linear combination of the alpha D glucose linked with the alpha linkage, and these of the linear compounds are water soluble. So amylose is a water soluble linear combination of the glucose, alpha D glucose, whereas the next one, that is the amylopectin, is the water insoluble cross linked structure of the glucose. When these of the two combines with each other, they form the starch. Starch is very essential in our daily life. They are the fuel for the human body. They provide the general daily routine energy for us. Now the second one, that is the cellulose. Cellulose is again made by the alpha D glucose, but here the linkage is the beta linkage. They are cross linked, they are beta linked, and they are generally available as a cell wall in the plant cells. What is the use? They work as a skeleton in these of the plant cells, and they are very important for the plants. But when you talk about the human beings are not capable to digest these of the materials, that is the cellulose. Why they are not capable? Because in our body an organ, that is the appendix, is not in working condition at the time, and as a result whatever the enzyme is needed for their digestion, that is the cellulose, is not available in the human body, and this is the reason human body is not capable to digest the cellulose in the body, and this is the reason why we can't graze. हम घास नहीं खा सकते, घास इसीलिए नहीं खा सकते क्योंकि हमारी बॉडी में सेल्यूलोज का फॉर्मेशन नहीं होता है. Now we are going to see the next compound, that is the amino acid. What are the amino acids? When we talk about the amino acid, we say that those are the compound which contains amino group as well as the acidic group are called the amino acids on the same carbon, that is the RCNH2, COOH and H. When you will see in the book, it is written when these of the group are not on the same carbon, even though they are called the amino acids. If they are on the second carbon, they are beta amino acid, third carbon gamma amino acid, but actually only the alpha amino acid, that means the amino acid in which both the groups are on the identical carbon, they are only of the biological importance, and this is the reason we say that this is the only amino acid, that means both the group must be on the identical carbon, then that will be called the amino acid. When we will see the nature of the amino acid, what is the nature of the amino acid, you will find that one group is acidic nature and another group is showing the basic nature. So what will be the overall nature of the compound? The overall nature of the compound will be the amphoteric nature, that can show the nature of the acid as well as they can show the nature of the base. So they are amphoteric by the nature, they are capable to show both the character, that is the acidic character as well as the basic character, and this is the reason amino acids are considered as a amphoteric substances. In this context, you can see its Zwitter ion nature. That means when you will see this compound, this group, a proton donor group, can easily provide the proton to this one, which is a proton acceptor, and as a result, whatever the species will be formed, that will be NH3 positive, COO negative, and this species is called Zwitter ion. An important topic, Zwitter ion, and each of the Zwitter ions are well known as internal salt. So they are the Zwitter ion or the internal salt, and this one is suggesting the amphoteric nature of the amino acids. Then how many types of the amino acids are? When you will see the type of the amino acid, you will find that there are two types of amino acids. The first one which are made inside our body, and second one which are not made inside our body. So those of the amino acids which are made inside our body, they are called non-essential amino acids. And those of the amino acids which are found inside the body, they are not found inside the body, they are called the essential amino acids. So which are found inside the body, they are the non-essential, and which are not found inside our body, that is called the essential amino acids. So there are two type of amino acids, and these of the amino acids are the monomers for the protein. They are responsible for the preparation of the protein by combining with each other. Here we can see that the loss of water is making a bond in between these of the amino acids. If these of the molecules will interact with each other, the loss of water takes place, and due to the loss of water, what happens? The bond in between these two will be formed, and this linkage is called the peptide linkage. So when the amino acids are combining with each other with the help of the peptide linkage, the polymerase chain of such species is called the protein. During the formation of protein, the sequence of protein is very important. Sequence of amino acid is very important. Whatever the sequence of amino acid is there, that will determine that what is the property of the protein. And for such determination, that is the determination of the sequence of the amino acid, it is the nucleus which is responsible. But the protein synthesis takes place in the ribosomes, which is situated at the endoplasmic reticulum. So from nucleus to the endoplasmic reticulum and ribosome, how the message comes? The message comes in the form of the codon. So the codons are the messages which suggest that which of the next amino acid will be linked in the chain. And in this way, the protein synthesis continues. Initially, the initiator codons comes and join with it, and as a result, the proceeding continues. The next codon comes for the next amino acid, next codon for the next amino acid, and again so. And finally a codon which is called the terminator codon, or sometimes it is called the nonsense codon. That means a codon which don't have any message for any of the amino acid comes and that terminates the chain. And in this way the overall protein synthesis takes place. So this is the overall function of the protein synthesis. Because these of the proteins are made by the big number of the such combination, that is the amino acid combination, which are made by the peptide linkage. So proteins are also called the polypeptides. Now when you will see that what is the structure of the protein and what is the type of protein, so we will see the structure and how these of the structures are going to be take place. As we knew that the proteins are the biggest molecule, we know, and if the molecules are such a big molecule, then there must be some arrangement of the monomers of this polymer, and that arrangement of the monomers is called the structure of protein. So we are going to see. Consider that these of the dots are the different amino acids. The first one, if we are considering the amino acid with the name of A, B, C, D, etc., then if the first one is A, second one is D, next one is Z, X, P, T, U, something like that, then whatever the sequence of amino acid is there in the protein, that sequence is called the primary structure of protein. So the linear sequence of the amino acid is called the primary structure of the protein. But I have already said that the proteins are the very bulky molecules. So there must be some arrangement of these of the molecules, these of the monomers. So the long chains will fold after a length. That means somewhat like this. And this primary folding of the protein is called a secondary structure. And after this folding, as the molecule is very big, after this folding, the refolding of the molecule will take place. And in this way, the molecule will be in this way. And so on. And this structure is called the tertiary structure of proteins. You can say that the secondary folding is called the tertiary structure of the protein. Now the final one, that is the quaternary structure of the protein. So this is a species. This was the primary, secondary and tertiary, and now we are trying the quaternary. And this structure is called the quaternary structure of the protein. So primary, secondary, tertiary and quaternary. The linear combination, the linear sequence of the amino acid is called the primary structure of amino acid. Primary structure of protein. The primary folding is called secondary structure, secondary folding is called tertiary structure and tertiary folding is called the quaternary structure. आप कह सकते हैं कि एक दूसरे में उलझे हुए से स्ट्रक्चर्स हैं, कुछ भी स्ट्रक्चर ड्रा करिए ऑलमोस्ट लगता है कि सही है. Since these are the structure of the protein, and on the basis of the structure of protein you can classify the proteins into two parts. The first type of protein is called the fibrous proteins. What are the fibrous proteins? The fibrous proteins are those of the proteins which contains only primary structure. That means they are linear. There is no any folding in the case of the fibrous protein. But if there is folding, if there is secondary, tertiary, quaternary, all the folding, then the protein will be called globular protein. Primary, secondary, tertiary and quaternary. All these structures are there. So they are called the globular protein. These of the proteins are also well known as native protein, that means the protein which are responsible for the biological property. So there are two types of the protein, that is the globular protein as well as the fibrous protein. There is another term that is called the denaturation of protein. What is the denaturation of protein? Sometimes in presence of some electrolyte, heat or some other factor, the globular protein turns in the form of the fibrous protein. And this phenomena of turning the globular protein in the form of the fibrous one is called the denaturation of protein. You have seen some of the example of the denaturation in your daily life. That is the boiling of egg. You have seen before the boiling of egg that remains in the form of liquid or semi-liquid you can say. But after boiling what happens? The system converts in the complete solid. There is not even a trace of liquid. कोई भी liquid जैसी चीज उसमें बचती नहीं. Then why such phenomena takes place? That is due to the denaturation of protein. The question is asked, where did the water goes after boiling the egg? Where did the liquid of the egg goes after boiling the egg? Actually that liquid, which is actually water, that liquid, that water is consumed in the process of the denaturation of the protein, because whatever the attachments are there for the coiling of the protein, that is the hydrogen bonding, and that hydrogen bonding attaches with the water molecule and that removes the water from the system, and this is the reason why it becomes solid. One more example of the denaturation of protein is the curding of milk. That means the conversion of milk in the form of the curd. That was called the denaturation of the protein. It's enough for today. Next day we will see the next topic.